Okay, good morning. Welcome to Pavitra classes. Today you are studying about one of the important topic in the biotechnology, that is the recombinant DNA technology. So, what is recombinant DNA technology? It is a form of artificial DNA. So, we are changing the DNA sequences. We are altering the DNA sequences by combining two or more sequences with the other organism of the relevant DNA. So other organism DNA we are injecting to the foreign DNA, we are injecting to the host cell, and we are changing the gene sequence. We are manipulating or alternating the gene sequence of the host cell, and we are again injecting it to the same host cell, and then they will be getting multiplication with the cloning procedures. The recombinant DNA technique was proposed by Peter Loban. This technique was realized by two scientists, that is the Loban and the Kaiser. Recombinant is a tool that helps in the understanding of the structure, function, and the regulations of the gene. So by adding the to add the other genes or other part of the gene, other organisms gene, we need to know all the structures and the function of the genes, including their products, including identifying the genes and isolating, modifying, and the re-expressing genes in other hosts and the organisms. So this is how the sugar phosphate backbone, this is the you can see here, this is the duplex model of the gene. This is the double helix structure of the gene. In between here base pairs will be there. This portion exactly it is viewed here. So this is the sugar phosphate line. This yellow line is called as the sugar phosphate line, and these are the nucleotide containing bases. So GCTA like this, it will be alternatively there. Gene modification of organism is done by the fusion of any DNA fragments to DNA molecules. So gene modification is done by other organisms DNA molecules with the DNA fragment able to maintain themselves autonomous replication. So it should be in the state. So other DNA molecule should be in the character that it should be having autonomous replication. Such molecules are called as replicants. So itself which molecule takes the replications, that is called as the replicon. So steps involved in the recombination DNA technology. So first the plasmid is cut with the restriction endonuclease. So its restriction endonuclease is the enzyme which is used in the cutting of the DNA or plasmid. Where exactly that cutting position is there, that sites are called as the restriction sites to produce with the DNA sticky ends. So two types of the ends are there. One is the sticky end and one is the blunt ends. The desired gene with the sticky end is to obtain human DNA by cutting DNA with the same restriction endonuclease. So again the desired other gene of the desired other organism gene, again that also cut with the sticky ends is obtained by the other human DNA. Then two fragments are annealed and covalently linked with the DNA ligase. So ligase is the enzyme which is used in as a joiner. Two fragments are analyzed. These two, first and two, second step where we cut the two parts of the one of the plasmid and other DNA part, that are annealed, covalently linked by the DNA ligase to produce the recombinant DNA. Once this joined recombinant DNA host by a noun process is called as transformation. So joining process of the DNA is called as the transformation. Then selection of the transformed host cell. So apart from that other cells, the selection of particular that transformed cell is selected. Then gene expressions of that transformed host cell and the multiplication of the DNA inserted host cell are done. Then again cell division will be takes place. This is the flow chart of the DNA in the bacteria cells in the simple general procedure. This is the cleared first DNA factor. First these restriction sites are removed here with the restriction endonuclease. Again these DNA fragments from other organisms, so they are removed by the restriction endonuclease. Then they are joined here like this. This is the recombinant DNA molecules, because this is the one organism plasmid and this DNA fragments from another organism. Then this is with the transformation procedure, this is injected to the host of this bacteria, Then growth and the cell division of the particular this transformed host cell is confirmed. Plasmid containing bacterium, this is once it is generated, then identical copies, that is cloning, is done. So identical copy of variety of the number of the identical copies, here you can see here, this is the number of the identical copies. When the plasmid containing bacterium with the recombinant DNA technology is generated, then identical copies are generated. This is the general outline of the recombinant DNA technology. Next is the applications of the recombinant DNA technology. So what are the application? Hormone production, in the process of hormone production due to progress in the recombinant DNA technology, bacterial such as cells such as Escherichia coli are employed for the fabrication of several chemicals and chemicals such as somatostatin, somatotropin, insulins and p-endorphins. These hormones will be prepared with the recombinant DNA technology. The concerned genes are integrated into bacterial cells which are cloned. These clones are called as are helped in the manufacturing of the insulin, especially insulin manufacturing, somatostatin, p-endorphin and somatotropin. These are the hormones can be production can be done by the recombinant DNA technology. Second one is the transgenic animal production. So biotechnologists have successfully created the transgenic sheep, pigs, cattle, rats with the help of the recombinant DNA technology. So significant usage of the transgenic animals in the manufacturing of the proteins as well as the some of the pharmaceutical composites also contributes in the function of the some of the animal species. Gene studies can be done with the transgenic animal production also. Also same with the transgenic plant production. With the help of the recombinant DNA technology, transgenic plants also generated improved properties such as the, especially in the plants properties such as resistance to the insects, herbicides, viruses. They also permit manufacturing of industrially significant biochemical and the pharmaceutical compounds. So with the transgenic plant production, whatever the manufacturing of industrially significant biochemical and pharmaceuticals which generated by the transgenic plants, that can also be production can be done. Next is the production of the antibiotics. So some of the antibiotics can be manufactured by the recombinant DNA technology. Example, especially penicillin, tetracycline, novomycin, streptomycin, bacitracin, etc. Next is the vaccine production. The vaccine are usually useful in contrast to the several diseases produced by bacteria. These are usually used as a may be killed and attenuated vaccines are used by bacteria, viruses and protozoa. These vaccines contains especially for malaria, polio, cholera, rabies, hepatitis and the smallpox. These vaccines production can be done with the recombinant DNA technology. To kill that organism and to attenuate that organism, recombinant DNA technology is used. Next is the biosynthesis of the interferons. So interferons are the have the antiviral and the anti-cancer properties. Usually virus infected cells will produce less interferon, but due to recombinant DNA technology, we can get the fair amount of the interferons. So with this procedure we will get the fair amount of the interferons which can be used in the which can have the good antiviral and the anti-cancer properties. Next application is the commercially significant chemical production. So recombinant DNA technology helps in the some of the manufacturing of the chemicals also, especially alcohols, alcoholic beverages and some of the organic acids, citric acid, acetic acid, vitamins are can also manufactured by with the recombinant DNA technology by the microbes. Next is the application of the enzyme engineering. So enzymes also play important role in the all the chemical reactions. Enzyme engineering can be clarified for the modification of the enzyme structure. So modification of the enzyme structure is based with the modification in the gene of that particular enzyme. So when the modification of the gene happens, then modification and alteration in the enzyme species also happens. Next application is the Prevention and the diagnosis of the disease. So recombinant DNA technology has rapidly expanded our ability to diagnose so many diseases such as AIDS, cholera, and genetic disorders also. Some hereditary diseases like Huntington disease, Tay-Sachs disease, some tumor cells, and monoclonal antibodies. Some procedures of monoclonal antibodies are also used to bear specificity of the contrary particular antigen. So particular antigen have the specificity of with the particular antibodies. So that monoclonal antibodies can be generated with the recombinant DNA technology. Last application is the gene therapy. So gene therapy is definitely a favorite area of the genetical engineering for human. It helps in the delivery of the particular gene into human body to correct the diseases. So some of the diseases can be corrected with the gene therapy if any damages are there. So insertion of the that particular healthy gene with the disease stated gene, to remove the disease stated gene and to help in the delivery with the good gene. That insertion method is helped with the recombinant DNA technology. So these are the 10 applications of the recombinant DNA technology. One is the hormone production, transgenic animal production, plant production, production of the antibiotics, vaccine productions, biosynthesis of the interferons, commercially significant chemical production, application of the enzyme engineering, prevention and diagnosis of the diseases, and the gene therapy. So this is about the recombinant DNA technology. What is recombinant DNA technology, steps involved, and the application of the recombinant DNA technology. Thank you.